This Capstone project focuses on bio-based production of adipic acid by Pratash Amin, Neil Chakravarti, and Huzefa Hussain. I'm Neil Chakravarti presenting this. Our advisor was Dr. James Kastner. So nylon 6,6 belongs to a family of synthetic polymers, and its cousin nylon 6 are the most commonly used nylons in the textile and thermoplastics industry. Their expected market value is 6.7 billion by 2026. It is known for having a high melting temperature, hydrophobicity, chemical resistance, elasticity, and washability. Nylon 6,6 is commonly found in textiles, but has also seen application in other products such as injection molding, 3D printing, auto parts such as radiator and tanks, and structural parts such as ball bearings and hinges. Nylon 6,6 is made as it is a combination of two 6-carbon compounds, HMDA and adipic acid. The reaction by which nylon 6,6 is synthesized is an equimolar polycondensation reaction shown below. A bio-based approach provides a sustainable and environmentally friendly to produce desirable petrol-based commodities such as adipic acid. Recent research into the use of genetically modified organisms such as strains of Saccharomyces cerevisiae and E. coli to produce precursors has shown promising results. A key precursor to adipic acid is cis-muconic acid, which can be converted to adipic acid using several different chemocatalytic processes. The benefits of this is non-toxic process, use of renewable feedstocks, and creation of byproducts for use in other industries. Our approach utilized a fed batch fermenter design. The parameters were controlled using PID controllers that would maintain the temperature at 37 degrees Celsius, a pH of 7, and a DO concentration of 20%. The use of metabolically engineered coli was used to modified form of the shikimate pathway to produce cis-muconic acid. E. coli cells will be grown using a glucose medium as well as a food source. There will be use of a palladium carbon catalyst to convert the muconic acid to adipic acid. This final conversion will take place in a PFR reactor for 2 hours at 25 degrees Celsius, 348 psi. We determined to produce roughly 55 million pounds a year of adipic acid in order to produce the 85 million pounds per year of nylon 6,6. From the data we gathered, this can be achieved if we used 15 250,000 liter reactors that run 364 days out of the year, with 1 day for maintenance and cleaning. Below shows our process flow diagram starting from the glucose all the way to the fermentation and finally the adipic acid. So looking at the biohazards and safety, everyone should be wearing PPE when handling any of the cells, the palladium, or chemicals. The waste flow is a mixture of bleach and water used to disinfect the reactor's interior. This would be a 10% bleach mixture with water, and any treatment before disposal should make sure that the waste flow is at a pH between 6 to 9. E. coli cells can be disposed of by mixing with soil and putting into landfills, and any use of third-party disposals should have permits and adhere to relevant 40 CRF regulations such as 403. These were taken from EPA guidelines. Looking over the final summary of our project, we would like to say that we do not believe that this process is entirely financially viable now, but with more research and advancements in technology, we believe this could be a viable option to replace current petrochemical methods used. Thank you so much.